Okay, so let's get started. So as usual, before introducing the speaker, just a few things. So you guys are all panelists, and which means you have the privilege of using microphone and camera. You can keep your cameras on throughout the lecture. That gives a more personal interaction with the speaker if you wish. But please turn off the mics during the seminar. If you have questions, as a panelist, you cannot use Q&A tab because you are a panelist. Those are for attendees, regular attendees. You have to use the chat box. You can take your question in the chat box, and then we can actually interrupt the speaker maybe once or twice during the talk, or preferably we can actually delegate all the questions toward the ends of the seminar. And now, I would try to actually invite you back to present your questions yourself, so you can actually really interact with the speaker a little bit better at the time. So I'm going to call on you with your questions. And with that, so let me actually proceed with introducing the speaker. So today's speaker is Professor Ulrich Kleinekathoefer from Jakobs University or Jacob University in Bremen, Germany. Ulrich received his diploma in physics from University of Göttingen, where he also did his PhD in physics, and then he did a habilitation in physics at Technical University of Chemnitz in 2002. And then after that, he joins actually the Bremen site of affairs actually as a research fellow at the International University of Bremen, which became actually Jakobs University due to the funding from the company. And then he went through the ranks there, and he's now a full professor of theoretical physics at Jakobs University in Bremen. Ulrich has been actually a close collaborator and colleague and friend of our group. He has been actually concentrating on applying combination of quantum mechanical calculations, classical calculations, and all sorts of other types of calculations to understand bioenergetic proteins that are involved in photosynthesis. Turn off your mic. Okay, I'll mute him myself, actually. So, and then we have been actually in touch. We have a lot of joint publications together, and he continues to be active in membranes and membrane protein systems, including actually over the last few years, concentrating on how antibiotics cross the membrane and reach the inside of the cell, which I believe might be something that he will talk about today. So it's good to have you back. Not quite, but electronically, Ulrich. It's nice to see you, and we all look forward to your seminar. Please.
Thank you for your invitation. It's really nice to see some old faces again, some old and new faces, and I hope we can make it somewhat interactive also in these interesting times. But I think it's very interesting to do it this way also. I mean, as Alec already mentioned, I just joined a presentation of Alec, which was organized by my colleague here and together with China. So it's getting really global these days. So it's very interesting. So let me start by sharing my screen, my presentation. Hope that works.
Perfect.
Okay. Yeah.
Yeah.
And let me just move over. I can see some of you. Okay, good. Okay, so so today I'm going to talk about the transport across bacterial membranes. As Emad was already mentioning, this is only one of my hobbies, I would say, which I developed after I came to Jakobs University at Bremen because of some experimental colleagues, especially Matthias Winterhalter, who does electrophysiology. But he only contacted me after he saw that actually I did some simulations of light harvesting systems together with Klaus. I mean, everything I learned about MD simulations I learned from your group. So that's the first thing. So if I did everything wrong, you can decide whose fault that is. Okay, but anyway, today I'm going to talk about membrane transport and basically mostly passive transport. I will start with a short introduction about the antimicrobial resistance. We will very briefly say something about the computational methods, just to be complete. Then I will just say a few words, how we start. Bioconductivity, something very simple, but in simulations still tricky sometimes. And then I will go into some different permeations of different antibiotics. Fosfomycin first is a very small one through a wide open pore, OmpF. I'll come to that. Then I will talk about the translocation through a much narrower pore from Pseudomonas aeruginosa, so OPO. And then I'm actually talking about some other larger antibiotics, Ciprofloxacin, Enrofloxacin. And if time permits, I will then also talk a little bit about ton B dependent transporters, which we worked on in the last years. Okay, let me just jump into this. So I guess I don't need to tell anybody that antimicrobial resistance is a big problem these days. Not yet as big as viruses right now, but maybe they can become such a big problem as well. So there's different mechanisms of resistance which you can develop, and one of the things is what you have to manage with antibiotics is to actually need to get into the bacteria, and that's why you have to permeate the membrane, and especially for gram-negative bacteria, that's actually a big problem. Then once you made it in, the antibiotics can be pumped out again, or your target might be modified and things like this. So there's a lot of tricky things, but today I'm basically going to concentrate on the influx. That is actually complicated enough by mainly beta barrel pores. Here's just a cartoon of the outer membrane, the envelope of gram-negative bacteria. They have an outer membrane and an inner membrane, opposed to gram-positive bacteria, which only have one membrane, but then they have an extra layer of thick layer of peptidoglycan. So today, I'm basically, as I said, I'm going to talk how the antibiotics go in. There might be hydrophobic ones, but they have lots of problems. So usually you try to have hydrophilic ones, and they need to go through pores usually. So that's why we're going to look at these pores, or porins often called. So there's a huge number, number of different pores in bacteria. There's those general diffusion channels, which are also called porins. They're pretty unspecific, and they actually restricted basically by the size of the substrates, something like below 600 Dalton. There's the outer membrane protein F, so OmpF, outer membrane protein C. So these are kind of toy models from E. coli. Then you have much more specific channels like OPRP, OPRO from Pseudomonas. And you might also have actually beta-dependent transporter for the influx. For the outflux, you will have other systems as well. As you can see, all these channels are usually all beta barrels with different number of beta sheets. They have lots of different properties. Let me just jump over this. They might actually have internally, many of them have a transverse electrical field, like the OmpF, OmpC. Some of them have positive charged ladders. Some of them have what you call a greasy ladder for certain special substances. Some, like the SIM-A, have a very uniform channel. Most of the pores actually are more like an hourglass shape, so they have a constriction zone in the middle, but some of them, very rarely, they also have this very uniform diameter. I started the whole business, or I was part at some point of this innovative medicines initiative, new drugs for bad bugs, and there was this special topic on translocation headed by Matthias Windhalter, my experimental colleague here at Jacobs University, and he is doing electrophysiology. there is the colleagues or some colleagues doing X-ray crystallography, and there we usually work together with Bert Vandenberg from Newcastle University in the UK. And we do the computer simulation, and I think these three techniques together actually can help you to understand much better many of the details, what's going on in the transport. I don't think in this audience I have to say anything about the MD simulations. I mean, our systems are standard systems, something like 100 to 150,000 atoms. I have to admit that for this system size on our local clusters, GROMACS runs a little faster, so that we use the GROMACS. And we use often to speed up simulation. So these antibiotics are they diffuse through the channel often. So these are transport times which are at least milliseconds. So we have to steer the process. And what we often do is we use so-called metadynamic simulations, where what is shown here in the movie, you basically fill up the valves and make it a flat landscape by inverting the blue parts, and you can actually read off the potential energy surface. So you just run through the configuration space, and basically wherever you have visited the place, you add an extra Gaussian potential, and then you fill up with this the landscape. I don't want to go into these technical details. What else we do steered MD, well known to your group. We also do applied field. I mean, I'm citing here Alex's work together with Klaus, because I still remember my first script to calculate ion transport I got from Alex. Thanks again for that, long time ago. And today I want to start very simple. Let's talk about, because that's something we revisited recently. Let's talk about this bulk ion transport, and just for fun, we wanted to know how good actually you can get these days. So here is actually conductivity. So you just have ions and water, and you apply a voltage across the simulation box. You can do the same thing, of course, in experiments. So this is at 1 molar KCl or 1 molar salt. The black curve is the experiment. The red curves are the standard charm 36 force fields. And then we played around with these recent Drude force fields. And let's and that maybe for now forget about the green curve, because that's kind of unusual high damping for the Drude model, which I would never recommend, but since they're also discussed in literature, we included them. So let's assume the blue curve. So you see the black curve is the experiment, and the red curve was the standard force fields, and the blue curves is a polarizable Drude force field. So for KCl, well, that looks quite nice, and the agreement increases nicely. Same for for sodium chloride, also there. I mean, the agreement, I mean, we're really talking now about quantitative agreement here. It's getting better already. The non-polarizable force field was okay, but you're getting better here. For magnesium chloride, the polarization does not increase. Polarizable force field does not actually increase here. Well, and for the calcium chloride, well, I think people will have to work on the parameters, because it's kind of disastrous how suddenly the biconductivity already gets. You can do the same thing now for the concentration dependence, and it's really astonishing how good the concentration experiment is now. If you have at small molarities, if you look at this blue and black curve, so experiment and Drude, how good they are for potassium chloride, sodium chloride. And also for magnesium chloride now, the range becomes much better. So you can go up to very high salt concentrations. Again, calcium chloride doesn't work. So we actually did this as a test just to figure out, I mean, what is about these polarizable force fields. But we then also tried to use this for ion conductance in the channel. And I'm not showing the results here because that would take too much time, but I have to say that we're, I would say, close to a disaster. So I cannot recommend currently to use polarizable force fields in the current force, the Drude force field for ion transport through channels. Bulks, they get much better, but already for potassium chloride, they became much worse. Okay, anyway, this was just a warm up. Let's talk about Fosfomycin through OmpF. So again, for me, I mean, as a physicist, I always have to learn what is my system at hand. So I first had to learn what is OmpF, so outer membrane protein F. So the nomenclature is pretty simple. So it's a homotrimer. So you basically have 3 channels together, bundled together always. So it's 16-stranded antiparallel beta sheets. And in the experiment, it has been shown that the pore can actually uptake Fosfomycin. So that's the antibiotic in use these days. And so here is what is Fosfomycin. So Fosfomycin on the left prohibits the so deleting or making MurA ineffective, and it inhibits the building of the peptidoglycan. There's a similar drug called Fosmidomycin, also with a phosphate group, and which I will talk about later on. We choose these 2 antibiotics because they're very small, and they have a phosphate group because later on I will tell you about the transport through phosphate selective channels. So then these should actually go through. So these antibiotics are in use for urinary tract infections and active against multi-drug resistant strains. Okay, nice thing, as I said, it's a very small drug, and the channel which we have here is pretty wide. So that's the widest channel you find basically in these bacterial systems. So it has a KCl conductance as 1 molar KCl of something like 4 nanosiemens, so it's pretty large. And also, if you take these small drug molecules, do an unbiased simulation, run for 2 microseconds, for example, you can see us on the right. So there's a kind of a trace to the on the horizontal axis, you see time, and then you see these traces, and there are 3 cases where the errors are where you get a complete translocation completely unbiased. So that is a very rare case that you can really see in an unbiased simulation translocation of an antibiotic, but usually we have to bias. And you can, of course, do all these nice movies now. You can look at interactions. Where is the Fosfomycin now interacting with which site group? And we actually came then to the idea, and also because our experiment colleagues wanted to do some tests here. What about if we start exchanging the positive? So the whole channel is hourglass shaped. So we are looking at the 5 residues which are the constriction zone. So in the middle of the channel, and they are basically concluding or so making the channel very narrow. And you see they are kind of on opposite sides. One is on loop L3, the other is on the opposing wall of the channel. And you see that they have they produce a pretty high electric field actually. So we thought, no, it's a negative. So Fosfomycin is a negative substrate. So if you replace these 3 positive amino acids, the arginines here, by neutral alanines or by negative residues, we should actually change the transport drastically, and that is what we wanted to see. And so it's a nice thing is always I can go to my colleague and say, okay, do this, please do this mutations and measure, and they actually do that. And so what we got, and when we did the calculation, so this is now based on metadynamic simulations, we calculate 3 energy barriers. So here the black curve would be the wild type. Then we have the blue curve for the AAA mutant and the red curve for the EEE mutant, which I was a little bit surprised about because I thought when I replace the 3 positive charges by neutral charges, I will see the barrier increase because it's a negative substrate. But then if I actually mutate again these 3 residues also to negative, I would actually have thought that the barrier in the channel would get even higher, but it almost didn't. You can now analyze the free energy surfaces in more dimensions by so-called reweighting. And then you can actually analyze this and see that the substrate is so small, it still can find its way through the channels without getting too close to the counteracting or to the amino acids with the opposite charge. So that was kind of surprising. What we did is this here, maybe as a kind of a technical point, we also developed some Brownian dynamics tools here, in that Brownian dynamics is usually used just for ion transport in these channels. So Benoit Roux, Van Til Im have developed certain programs long time ago. We extended these programs to actually be able to include parts of the simulation of the system in all atom. So we can have the substrate, for example, in all atom, or we can actually also have a loop in the system in all atom. Also, the scaling, I have to admit, with the number of explicit atoms is pretty bad, but still, if you don't have too many atoms explicitly, that's not so bad. And also you get the electrostatics much better because you have an explicit representation of the atoms, and larger molecules actually can also then wiggle through the channel and things like this. So we combined this, we developed this, and we also had actually to, I mean, it's not so simple what kind of diffusion constants you have to take, because now the water is implicit in these models, and suddenly you have to actually map this fully atomistic description together with this implicit description. So we had to come up with some kind of diffusion scalings and things like this. So, but that is more a technical point. What we got, what I found very interesting is if you now look at the flux rates as in terms of molecules per second. So really flux molecules per second is if you do MD simulations at a rather large voltage, 1 volt, so it's larger than you have in experiments. Usually you get something like 100 times 10 to the 4. And of course, its barriers of AAA and EEE is somewhat higher. I mean, the flux rates are a little bit smaller there. We can do the same thing in our reduced BD model. So we get very similar numbers. So they're off by a factor of 2, but that's actually not. And now do a much longer simulation. We can also calculate the equilibrium crossing rate without bias, or we can actually calculate at small voltages and compare to what I call experiment-based. I cannot really, I mean, there is an assumption how you extract these numbers from the experiment, because these are not direct electrophysiology measurements, but these are so-called reversal potential measurements. But as you see, the numbers are all in the same ballpark. So it's 10 to the 4, 10 to the 5 molecules per second. So I find this very intriguing that we actually get numbers which are in the correct ballparks. Also, if you talk about actual translocation numbers, which is not so trivial, because if you get the potentials wrong in a way, they enter usually the rates exponentially. Okay, so this was the simplest antibiotic. Let's go a little bit more complex. Let's look at the different channel. Let's look at this OPAO channel. So that's now a channel from Pseudomonas aeruginosa. So it's a pretty nasty bug, and you have to understand that these these organisms, they don't have these large pores like in E. coli. In E. coli, I just was showing this OmpF channel, this rather large channel. These bugs don't have these large channels. They just have very narrow, very specific channel. And this OPAO is expressed under phosphate starvation conditions and is phosphate selective. Actually, the OPAO is for diphosphate, and there's a very similar system which is called OPRP, which is more selective for monophosphate. Anyway, and the idea was, okay, if these channels are selective for monophosphate and we want to get something into this bacterium, so we want to use this channel, so we want to use some drug which also has a phosphate group. So the idea is that actually these can then enter through the same mechanism. So we took this Fosfomycin, so this was this other phosphate containing drug which is in use. And we looked at how we can get this through. So as I showed you earlier, we used to do always our simulation with metadynamics. And in the computational field, of course, many people and also I guess many of you will have used umbrella sampling also. And people kept telling us we should use umbrella sampling. And I was kind of a little bit skeptical for some reason. But then I would say, okay, let's try out what we get. So the nice thing here is, since it's a trimer, so the pore, here's a trimer, we can do 3 simulations at the same time. So we get actually a tiny bit of statistics, only 3, but still. So we redo when we calculate the free energy surfaces 3 times at the same time. So we do this with metadynamics, we do this with umbrella sampling. They look very similar, and the averages you can see here. I mean, I think this is what you would expect with 2 different methods. But this is, okay, I have to specify this. This is only a chloride ion right now. Okay, we are looking at the translocation of 1 single chloride ion here. So everything looks fine. We take these 2 different methods. It's, as I said, it's a phosphate specific channel. So it's specific for negative, it's for negative substrates. So it's no wonder that also for chloride, you find these binding spots in the channel. And you now can analyze the actual position. I will come a little bit to this later on. As a next step, we made the substrate a little bit larger. So we now took a monophosphate, so H2PO4 minus molecule. So we look at the distribution of now the PMFs. So, I mean, they get now not so nicely agreeing anymore. In case of umbrella sampling, you even get this kind of barriers here. Look at the averages, so it's kind of still okay. But the agreement gets worse before interpreting, I mean, any physical details here. And the worst thing comes if you go to really our antibiotics molecule now. For the metadynamics, the results looks still reasonable. For the umbrella sampling, if you take the blue curves, the umbrella sampling and non-periodic boundary conditions, you get the blue curves, which are kind of very bad. But you unfortunately see this over and over in literature for these channels. So what happens, I mean, the PMF at the two sides of the channel in bulk, they should have the same value, of course. I mean, you enter the channel, there's a barrier or well depth, and you leave the channel again. Now on the other side of the channel, the energy should be the same. And there's lots of problems with this. People often use the so-called umbrella sampling with periodic boundary conditions, but that's kind of swiping the problem onto the carpet, because you enforce this periodicity at both ends of the channel, although you see there's something very problematic going on. So I cannot really recommend this. And of course, I know now you will say, well, you didn't do enough windows and all these kind of things. That's not the case. You would say, okay, do replica exchange, umbrella sampling. Actually, our collaborator Im actually said this, and then they did it, and they get still stuck. You know, they still have problems with both sides. So it's not a simple problem to solve in umbrella sampling. So especially for larger substrates, there is, I think there's a really problem with the VAM algorithm here. Anyway, let me just jump to the movies. I mean, or the physical interpretation of the whole thing. Now, if we have now our Fosfomycin molecules, of course, you can see them going through the channel, and now it's a narrow channel. And you can see it's on the left panel here, you can see the path in a free energy scale. So on the x-axis, you have the channel direction, so this is Z, so that's the channel direction here. And on the other direction, you have an orientation. It's not actually, it's something you can interpret as the orientation of the molecule. And one of these questions the experimentalists always ask, what is the, if going to the constriction zone, which part of the molecule goes first? So which part are we not allowed to modify too much so that we don't disturb the permeation through the channel? And then you can, for this thing, you can also do from the, you can do another simulation with the other side. So So with the other side of the molecule facing ahead. And actually in this picture here now, you see the channel now, it has a different orientation while going through the constriction zone. Maybe I just show both at the same time. So one channel, once it's going up here, so that's the phosphate group ahead, and the other one is going down here, so it's the nitrogen group ahead. In the middle, there's a large barrier, which means actually the molecule cannot go perpendicular to the channel when you go through the constriction zone. What is plotted down here is now the energy if you go along these paths, and you can go along path 1 or path 2, and there's a little higher barrier for path 2, but the difference is actually not so big. So you can analyze this now in detail, but this, as I said, is a very small drug, but it's in principle should go through this, and the free energy barriers are something like 5-6 kcal per mole, so it should be still able to permeate in reality. That, I mean, that's our experience, what we have so far. Let's go to some larger substrates. Things get really tricky then. Let's, for a change, go back to larger pores again, because otherwise you don't have any chance. So these are very standard drugs. Ciprofloxacin, I mean, you might have taken it for, 2-3 years ago on a vacation, I had to take it because of an ear infection and things like this. So that's a standard antibiotic you will get prescribed. OmpC is a smaller brother or sister of OmpC, so it's very OmpF. It's very similar like OmpF, just a tiny bit smaller. It is, we actually studied 2 different drugs. We looked at the ciprofloxacin and we looked at the enrofloxacin, where you see the only difference between these 2 molecules is that there's an extra ethyl group here. Other than this, they are similar, very similar. The nice thing is also that there are electrophysiology experiments which tell us how many molecules go through. And for ciprofloxacin, it's something like 3 molecules per second, enrofloxacin, half a molecule per second. Just keep in mind for the Fosfomycin through OmpF, we had rates of something like 10 to the 4 molecules per second. Okay, so the rates there were much, much higher. So we are really, these are larger compounds getting into, and you need these large channels so that you get in. We actually wanted to be, because in this business, you see lots of calculations, and there's lots of calculations I don't trust too much. So we want to be very careful in actually calculating the free energy surfaces. We actually did it 3 times. So we always look at 2 CVs. We look at the distance, so the channel axis, and the other one is the orientation of the molecule within the channel. Actually, we never really managed to calculate a PMF by steering both at the same time in metadynamics, but at the end, we only steer one and we calculate the other one by the so-called reweighting procedure. That is shown in the next picture. So you see 3 sets of free energy profiles here with slightly different steering parameters. In principle, I mean, the results should be more or less independent of the steering parameters, at least in a certain range of steering parameters. It's very realistic. What you can see here, so again, this is the channel axis, so the molecule is going through this channel, and this is the orientation. You see there, so it's a little bit hard to see, there's a black line here, so the molecule can basically, this is showing the rotation of the molecule while it's actually going through the channel. It's always going on at this side here. It's always going with an amine group ahead. You see the PMFs, the 3 PMFs are not identical, especially not in these regions where which we did not steer. But if you now look at the free energies, if you go along these paths, for these 3 samples here, then that is plotted here. So you get these 3 different PMFs projected onto the 1 dimension, onto this direction here. You see the results are very similar, not identical, but very similar. And actually, we really wanted to be sure that what we see here as minima, so we identified 8 minima, are really minima. Because sometimes in these very fancy simulations, I mean, lots of these metadynamic simulations you find are being done for toy models. So this is a really large system with lots of problems in convergence, so really try to take care. But then we really wanted to make sure that the minima are actual minima. So what we did, let me just jump about this for a second. The minima, so what we did, actually, we identified the minima, put the molecule in the proper position and the proper orientation, and let it run for a couple of 4 times 25 nanoseconds, or sometimes also actually somewhat longer. And if you're in the minimum and to do an unbiased simulation, you should not actually run away from that position too quickly. And that is actually what happened. So if you do this, the simulation actually they look for the same areas. This is kind of, if you map this onto the PMFs, this is actually very much what you find from the PMFs. So the minima we found in our simulations are really minima. So that sounds trivial, but with these kind of complex simulations, unfortunately, that is not always the case. Now we can actually analyze what does it mean for the molecule to actually be at this position, then and this one, and go through the channel. But I can also show you the movie. So one is from the side and one is from the top. So you see the molecule getting in with one orientation. Then actually it has to turn around. And then it comes to this constriction zone, this very narrow part of the channel. And the residues we show here in green are all those for which we found a reasonable interaction in the metadynamic simulation. So this is based on a forced simulation, of course. In unbiased simulation, you would not see any translocation. So this is built out of our metadynamics trajectories. But it gives you a good idea so what is happening, and also, as I said, this turning around and all these kind of things we can see in the PMFs as well. But this gives you, of course, a nice idea of what is happening. And now, based on these schemes, you can now try to analyze what is going on and make suggestions what you might want to change. And as I said, there was this other molecule, Enrofloxacin, which only differs by this ethyl group. So we did the same thing. We calculated the PMFs. We calculate then the free energy along this minimum energy pathway, and get the blue curves. And the ciprofloxacin one is this orange one here. And you see the barrier is higher for the enrofloxacin than for the ciprofloxacin, which is actually very nice in agreement with the electrophysiology experiments that the rates of ciprofloxacin are higher than those of enrofloxacin, for which the barrier is higher. So this is fully consistent with experiment what we did here. But of course, these simulations, I mean, basically these simulations for enrofloxacin and ciprofloxacin more or less took one student. I mean, that was a whole PhD thesis basically to get it converged. We tried out a lot of things. We had lots of failures with certain methods, but at the end it worked out. Okay.
So can I ask a question?
Sure.
So in this, you said that you are taking one collective variable for the metadynamics, and then the rest you just sample and analyze later. So is this the translocation that you always take as the main collective variable? As the
Yes.
Okay. And then actually the second thing I was going to say, for the second drug, the enrofloxacin, does it take the same path, like the same kind of?
More or less.
More or less.
So, I mean, you can see, I mean, especially, as I said, what the main question you always get from the experimentalists is which orientation it goes through the constriction zone. So for the other one, for the ciprofloxacin, I mean, this is one way, and the other orientation would be going up here.
Yeah.
You see, both of them are very similar in shape, basically. Both times the orientation is the same. If we take, I mean, we have actually now a case for a different channel where this is not so clear anymore. So then you'll get get kind of over the transitions on this side here.
Okay.
But that is always a main question. If you talk to people, and that was a nice thing about this IMI project and translocation, we worked together. We talked to people from AstraZeneca and Glaxo and people like this, and that was their main question. What is the orientation it goes through?
Yeah.
What are the rough rates?
Absolutely.
But of course, for them, this kind of simulation was much too slow. I mean, as I said, this was a PhD thesis. German PhD thesis takes like 3 to 4 years. So one drug per 3 to 4 years is not what they will accept. They talk about 1,000 per day. But anyway, so what we did for this case. I don't show the data now. We also did this Brownian dynamic simulations here with these drugs with the same channel. Of course, you don't get the fine details of the PMFs and when we do this Brownian dynamics together with some kind of temperature acceleration, but we can actually reproduce the fact that Enrofloxacin has a higher barrier than Ciprofloxacin, and we can actually reproduce rough barriers, which is more than actually the experiment than I was expecting initially, and would be actually nice for a fast pre-screening maybe. Not to get all the fine details, but sometimes you need a fast method. And this Brownian dynamic simulation for getting PMS was a factor like 5,000 faster than this all atom. Anyway, so
on that metadynamics plot, if you could go to the previous slide. So this white space.
Sorry, yeah.
Not this one. The metadynamics, the 2D profile.
This one.
Yeah. So this white space means what? There was no sample here, or
yes. So the as I said, I mean, we only steer along the z-axis, and the other directions, like the orientation, which I show here, is only sampled without steering.
I see.
That's why you don't reach all the. I mean, these are higher energy areas where the system doesn't like to go. In this example, for example, it somehow samples this area for some reason. But our strategy here is that you will, I mean, you will sample the orientations which should be close to those, I mean, those you need in a way. I mean, we're also trying, I mean, this is kind of higher dimension. Of course, we would like to do higher dimensions. We're actually trying out something like this, what's called the method TASS, the temperature accelerated slicing by Nishant and Nair with 8 dimensions. Yeah, but this is very tricky. I mean, this is the problem is of at least steering one dimension, but then you get these empty white spaces. But we can discuss this later on.
Actually, really, sorry. Oh, sorry. Are you still answering the question?
No, go ahead.
I'm sorry. I'll interrupt you because I really want to know your answer to this question, but I have to run at 10, and many people have the same question. So, what is wrong with umbrella sampling? You know, we all do the simulations. We all see this tail, you know, on the other side, and it's really annoying. So, do you have an idea?
We are looking, I mean, what we now saw, I mean, as I said, this Nishant Nair, we are doing this this temporal accelerated slicing business. And what we had, I mean, huge differences. And
yeah,
now,
I mean, it's the same thing. It's we just never publish it.
Yeah,
but
in my feeling right now, it's the VAM algorithm. We are trying to do something with slicing the different pictures together with different methods. So now we're trying some mean average.
You know, you always you kind of see it when you have a membrane. Like if you don't have a membrane, it's fine, you know. So I think one explanation, I think, is from GC that it's very slow to equilibrate. You know, okay. Yes. I mean that's what we thought. You know, is the reason. But you're saying if you do a different method, it works better.
Yeah, it certainly works better. It depends on the method.
Correctly.
But of course, it's all a problem of, you know, we are only doing 1 or 2 degrees of freedom here, and it's a very high dimensional problem. And I mean, that's why I did this earlier example. I mean, as long as you do chloride, everything seems to be okay. You know, you increase the complexity of the substrate with monophosphate, it starts to develop, and then you get an even larger substrate where you need more time to equilibrate, and all the other degrees of freedom come into play. Then it actually gets really bad. So yeah, it's but we also tried. I mean, we actually tried hard to take more windows. We run longer per window and all these kind of things. And as I said, one pill actually couldn't believe it and said, okay, I do replica exchange. And 2 weeks ago, I asked him again, so what about your replica exchange? He said, yeah, we don't have it.
We tried that too. It's same thing. Yeah.
Yeah. So metadynamics in this aspect is much better. Part of the story is also that it's the substrate can actually go back and forth. So it can go back, I think, from the barrier region, can equilibrate a little bit and can go.
Kind of samples deeper in the membrane, for example, if you were to use analogy, right?
Yeah, exactly. So,
okay.
But this is something which really is annoying. And I mean, always I see these papers where people publish this and then they do this kind of periodic boundary condition business. Yeah. And I think that is kind of cheating. I mean, you kind of just force it away, you know, that I don't like. Anyway, we can discuss more later. Yeah, I mean, of course, you can now with the Ciprofloxacin and the Enrofloxacin, with these two different, very slightly different drugs, you can, of course, now start and analyze where the difference really comes from. Why is the drug, I mean, where's the difference coming from? And then you can see that there's a hydrophobic pocket for the Enrofloxacin interactions here. But it's a poorer, but here you have a charged interaction. You can analyze this, and this is the kind of details at the end, if you talk to pharma people. That's what they want to understand, how they would have to modify a certain side group of their drug to increase permeability, because permeability of the antibiotics is a major problem. If you ask the people in GSK, they will tell you we have 10,000 compounds which would kill the bacterium if we get it inside, but they don't get them inside. So if they want really to understand which kind of side groups you have to modify and things like this. So what is kind of general rules? Okay, let me give you very quick, I mean, now I'm kind of running out of time. Quickly about a little bit about ton B dependent transporters. And this is a work, I mean, mainly driven by the crystallographer Bert Vandenberg, and also only because there's a nice crystal structure, you get it published in Nature. And recently we had an update on a slightly different, so the first paper was on SUS-CD and the second on RagAB. So what are these? So these are actually human gut bacteria. So not all bacteria are bad. You don't want to kill all of them. So in your gut, you don't want to kill them. And there's this starch utilization system, SUS. And there's a whole cascade, as usual, whole machinery of different proteins. So there's starch, then it's cut into pieces. And then comes the SUS-C with actually SUS-CD part, which brings it across the membrane. And this is ton B dependent, so there's a plug inside which has to be pulled out before actually can go in. We focused basically only on that first part, so basically we only looked at the SUS-C and SUS-D part. So the point was that Bert Vandenberg came up with this crystal structure. So what is this? So the yellow part, and actually it's a dimer, so oligomer. So the yellow part is the beta barrel. Then there is this reddish part, which is closing the beta barrel, so it's like a cap. And in this crystal structures, he always had this peptide bond. So without a peptide bond, he could not actually crystallize this D part. So the C part was basically known, but not as a whole complex. So now the question was, so what happens if you take the peptide away? And they could not resolve this in crystal structure. He came to us and asked us to take it away. So left is with the peptide and here without the peptide. You see, actually the cap actually opens automatically. We have to admit, we have a little bit lucky here. Let me just do it again. So that in this, I mean, this opens spontaneously. So if there's no substrate bound, so that's pretty nice. Of course, I mean, at least this was kind of in one of the early simulations. So we were kind of seeing what roughly goes on. So we actually could do more. And then the problem, of course, was also that, you know, there's this, it's a dimer, and there's not only 2 beta barrels and 2 caps, but there's also 2 different kinds of lipoproteins, which are associated with it, which are also resolved. So we had to put everything into the simulation box because one of the questions also of the referees at the end was, are these 2 caps hindering each other while opening? And well, we have to simulate this. And luckily, again, in this simulation, one of the caps actually opened, which was very, very nice here. And we're lucky in a way, but we repeated many simulations. On the right-hand side, you see actually this peptide bound. I mean, the cap, this is the distance between, you know, the beta barrel and the tip of the cap or the lid, and it stays always closed. And if you remove the peptide, there are different kind of simulation conditions, but in most of them, you see a wide opening. And of course, it takes also quite a while. So this 500 nanoseconds business, at least, you know, and it's those which don't open yet. I mean, they might open later on. This we cannot say. It's unbiased, completely unbiased simulations. Okay, so that's, we are very, very lucky here. I have to admit that we were really lucky in that Bernd van den Berg later on gave us the same thing, the same structure for a different organism, which actually has a few more salt bridges between the lid and the beta barrel. And there you don't see anything opening spontaneously. There you have to actually force. And this came up then with these pedal bin mechanisms. So what happens? So you have the beta barrel, you have the lid, and once, you know, this peptide comes, binds to the lid, then the lid is always fluctuating a little bit, and when the peptide is bound, it will actually flap closed. And when it's closed, the peptide can actually connect to the plug domain, and then you can actually steer it out. But this part, we are still actually looking into how this really works. There's different ton-B dependent transporters, and this is interesting how different they are. So this was kind of the idea of what really happens there. Recently, there was another organism. This time it's not a gut bacterium, but this is actually causing human periodontitis. Yeah, also something you don't want to have. It has a very similar structure, so it's RagAB there. The difference now was in this new publication. Now there were collaborators doing cryo-EM, and with cryo-EM you could also resolve open structures of the whole thing. So what is actually shown here is in green, and on the right hand side, in the green structures, our most open, wide open structures we get in our simulations, while the red ones or pinkish ones are the cryo-EM structures. So interestingly, we don't get the fully open structure, at least not in unbiased simulations. And even so, what is shown here with the RMSD business here, basically the green ones are with peptide again, so they stay closed. The red ones are without peptide, and they start to open to a certain degree. And then there are blue curves where you start with the MD simulation and the open curves. And you start, they go to some structure in between, so to say, between open and closed. It's basically, it's a lid opening, and then the lid turns around to the side a little bit. And this turning to the side, we don't see in the simulation. Interestingly also, these structures, what we see is very similar to what we saw for the previous complexes, so CD. So for some reason, we don't see this last part of this opening mechanism, or that part is artificial for the, it's a crystal, or it's a cryo-EM artifact. That is so far a little bit unclear, but this structure is very similar to what we had before. So the interesting thing is that now many more of these structures are getting resolved from human so which are really important for humans from these bacteria, which people were not able to resolve earlier because these bacteria apparently are hard to handle, which I cannot say anything about, of course. Okay, so with this, I think it's time to close here. Oh yeah, one more slide. So I didn't talk we also did, for example, simulation with LPS membrane. So this is simulation collaboration with 1PIL-IM. So we had some ion transport to one of these channels and together with LPS. We also looked at a mechanism for the maintenance of lipid asymmetry. Again, this is a project together with the crystallographer Bert Vandenberg. So there's an interesting mechanism which does not need any energy to bring phospholipids from the outer leaflet in these outer membranes back to the inner side where they are picked up by a different protein, because the outer side of bacteria, outer membranes, the outer leaflet of the outer membrane consists of LPS, and the phospholipids or rafts of phospholipids can actually endanger the bacteria. With this, I would like to actually stop, or just, okay, as a summary, it just basically is a copy of the introduction. So I talked a little bit about the bioconductivity, showed you that the agreement with experiment already for bioconductivity is good, but there's still issues with force fields, especially also if you want to go think, okay, just take polarizable force fields, the business can be slightly tricky. I talked about this Fosfomycin, this very small drug going through a very wide channel. So again, this has a conductance, KCl conductance of something like 4 nanosiemens. This channel from Pseudomonas then has only a conductance of something like 250 picosiemens, so like a tenth of it, roughly. And then I talked about these larger substrates and showed you these translocation of those. And then at the end, I showed you a little bit about ton-dependent transporters. And of course, here's a group photo. I should actually get a newer photo, but these days with social distancing, you don't get a nice group photo. So here is, for example, Jignesh did the Ciprofloxacin and Enrofloxacin business. Jignesh did the Fosfomycin and Fosmidomycin business. I think the experimentalists, Matthias Winterhalter, Roland Benz, were a very fruitful collaborations at our university. Bert Vandenberg, our friend for crystal structures. With 1PIL, we worked on the LPS, and I thank you for your kind attention. Thanks.
Well, thank you very much, Ulrich. Very nice, very stimulating conversation and presentation. We have a lot of questions. I'm going to actually invite the audience to sort of ask their questions themselves. Claudia, you want to go first?
There you are.
There you are.
Hi. So, I mean, my question has to do more with biology, and I should know the answer, but I don't. So probably you do. So these porins allow antibiotics to cross the outer membrane, but yes, not all. But some of these antibiotics still need to cross the inner membrane because they act at the level of ribosomes or something that happens inside the cell. Mm-hmm. So do we know how they gain access to the cytosol, to the cytoplasm of the bacterial cell?
Yeah, I mean, for some, I mean, it's very specific then for the individual cases. For the Fosfomycin, Fosmidomycin, for example, it's known that there's a transporter active. I mean, it's the inner membrane, then you don't have these big water-filled channels anymore in the inner membrane, because, sure, I mean, it would kill the
Of course, no, right. That's why I'm asking. Yeah.
Yeah, that's. I mean, there is. I mean, they have to be picked up by transporters, and that's the next thing. Then we also. I mean, for the Fosfomycin, we looked into this. There's one of these transporters which are probably
okay, secondary active transport.
Yeah. It's GLPT actually. GLPT is specifically for Fosfomycin.
Yeah, exactly. I was looking for the name. Yeah, GLPT.
Yeah.
And you worked on that.
Yeah.
So this we looked into this at some point, but it's it's very time consuming. to do
Cool.
this. Yeah.
So Andrew Peng, are you still around? You had a question about Andrew Peng has a question about well-tempered metadynamics. Are you using well-tempered metadynamics?
Yes.
And how long is the metadynamics and umbrella sampling simulations?
Yes, I mean, I didn't say too much about the technical details. So yes, we use well-tempered multiple Walker metadynamics. So usually we use something like 16 Walkers, so 8 on both sides. The simulation and total simulation time is roughly 4 microseconds. What we did also for the umbrella sampling. So the comparison I showed for the metadynamics umbrella sampling, actually we had at the end much more time spent on the umbrella sampling, although the results look much worse. But I mean, as I said, we only steered in one direction. Maybe I should quickly go back to one of these PMFs, because that is of course always one of these questions. So we only steered along this direction. So we actually, of course, tried to also steer it to get a full two-dimensional PMF. We gave up at 16 microseconds. And I was asking the poor student always, so which is the orientation of the drug which it goes through first? And the answer would change every week until I stopped, because I mean, it was really, I mean, every week, I mean, there was still, the thing was still changing. So convergence was a huge problem. That's why we gave up. And I think that's this steering one dimension and doing this reweighting in the other direction. I mean, the philosophy is that for the other degrees of freedom, you're hopefully, I mean, sampling close to those configurations at their minimum, so to say, in the other degrees of freedom. Maybe not the exact one, but I mean, that's why we then get these ranges around this minima, and then we calculate the minimum energy pathways. And you see, usually we do have some sampling around these pathways. Of course, there's always a risk that we miss something. And we are trying, I mean, we have now this temperature accelerated slicing method where we try to force 8 degrees of freedom. The sampling is much better, but then there's other issues.
Jale, you have a question which was also my question. So you want to ask your question, Jale?
Yeah, I think you just kind of answered. I was wondering what was wrong with the 2D. So you just said that it never got converged when you were biasing along the orientation degree of freedom?
Yes, exactly. So
it
never got converged. And I mean, we try, I mean, the convergence is really a big issue. I mean, there is some, if you look for metadynamics, there's some recipes of convergence of error measures. But even if the error measures are all fulfilled, it looks good according to those measures. And then you do the same simulation 2 or 3 times, you might get completely different answers. And that's why we also then said, okay, let's do, that's what we usually now, I mean, that's what we did here in this simulation. We did the same simulation 3 times with slightly different steering parameters just to be sure we cannot reproduce the results. Because that is I mean, yeah, as I said, I mean, that was a painful way. So, I mean, and they don't, I mean, they're not 100% the same, but.
Oh yeah, definitely. Because in the movie, you show that initially there is a flip, and then the drug goes on in some other orientation that you originally.
Yes, it starts here at number 1. Yeah, it's this picture. It starts number 1.
Yeah.
And then to number 2 means it basically starts to rotate. And number 1 and number 3 are basically opposite orientations of the molecule. That's exactly what you then see in the movie. It goes in this direction, and then it starts to actually turn around. And that is what you can also then read off from the PMF. So then it's turns around. And so this initial part is really going from number 1 to number 3 here.
Correct. Yeah, I guess that in the constricted region, then there is no change in the orientation. So that was, I was curious to know why it wasn't getting converged, because it seems that the bulk part of it determines what orientation it goes in. determines it at the larger radius of the poring, right?
Yeah.
Okay,
The point is, of course, if you start to steer the second direction, I mean, there's a huge barrier in the middle.
Yes.
And
yes.
if you start to steer in that direction, you try to, sample that huge barrier. I mean, depending on, of course, on the steering parameters, but this causes tremendous problems to our. I mean, that's what we saw. Yeah.
Okay. Yeah, I mean.
Go ahead, Charlie. Finish
I was just wondering if you must have heard about the path collective variables. Do you think that path collective variables now could be used along this path that you found? Because path collective variables give the opportunity to be 2D along a given path, and then one can sample along the orthogonal degrees of freedom a little bit.
Yeah. It might work. We did not try it out. We did not. We did not try it out. It's very hard to predict with these methods, I have to admit.
So apparently, JC is with us, and he would like to comment on the differences between umbrella sampling and metadynamics. JC.
Yeah. So hey, Ulrich, it's good to see you again and see everybody. So regarding the error that you found in umbrella sampling, so we also looked at this, and I'll send a paper in the chat a few years ago. And the reason is essentially you probably use something like steered molecular dynamics to generate your starting states for umbrella sampling,
yes,
as most people do. So you're accumulating non-equilibrium error essentially as you pull it through. Because you can never pull it slow enough, right? So, and then when you run umbrella sampling, you're asking the algorithm to essentially equilibrate all the windows individually. So we did the same. We did this for urea going through a membrane, and even after 4 microseconds of total sampling, we still had an error. Okay, so then why does well, I'll say one more thing about that. So then when we equilibrated the windows longer, before starting the umbrella sampling, the error was greatly reduced. And so it was clearly these non-equilibrium effects. So, okay, so why does metadynamics do a better job? We saw the same thing with ABF, adaptive biasing forces, that it did a much better job with regards to this periodicity issue. And the reason is that essentially the ABF or metadynamics algorithm is slower in terms of how it explores the new space. So instead of a 50 nanosecond or whatever SMD simulation, you're giving it microseconds to explore the space at the same time it's calculating the free energy.
Mm-hmm.
Does that make sense?
That is part of the answer, yeah. But
I agree, JC. So because actually we are spending a lot of time manually making sure that the two windows connected kind of before we sort of even actually exploring all possible orientation in two different dimensions, just to make sure that you have a connected kind of path before you go in and do the umbrella. I think the initial seeding can make a huge difference.
Important. Yeah, exactly. And
I completely agree with that. I mean, in a way, this study was basically, I have to admit, was forced by me being kind of annoyed of always saying, okay, you need to do, by colleagues here in Germany, always saying, okay, why don't you do umbrella sampling? It's so simple. And then I see their papers publishing these things, they publish. And you see, I mean, this kind of, you see, I mean, if you go to the literature, you find a lot of these reports. If you, I mean, then they do periodicity. There is, I mean, I agree. I mean, if you probably, I agree with JC, probably it has to be something like this, that your initial windows, we played around with this, we tried to take care, but probably not enough, because we're also not experts in umbrella sampling.
Sure.
But yeah, it is a major.
Yeah, so I don't think, but I guess just to summarize, it's not a problem with WAM or the algorithm. If anything, it's being very honest with you, showing you the error that you've accumulated, but it can be dealt with if set up carefully.
Can I ask
one more question, and then we have to move to our kind of private meeting with the speaker. during which you guys can continue the discussion. Go ahead, Nandan.
So when you did the SMD during umbrella sampling, did you restrain the protein? Because if when you pull this drug, the protein might be completely disrupted. That might lead to the non-equilibrium effect. Like,
no, we did not restrain the protein, but I think, I mean, we did different pulling speeds and all these things. We also tried to get some windows from the other side. You also have to be very careful that actually you need two. I mean, for larger drugs, I mean, in the channel, the drug cannot reorient anymore. So actually you will have to, in umbrella sampling, will have to calculate two pathways in the constriction zone separately. There is all kind of issues. So we tried a lot of things, but probably JC is correct that we are still not careful enough. Yeah, but it's, but I think for an umbrella sampling, we tried already a lot of things, and the simulation time for this was already much higher than for the metadynamics. Yeah.
Okay, cool. Thank you very much, Ulrich. This was really stimulating, very nice. Thanks for sharing all of your experience in this difficult, complicated molecular system with us. So we are all struggling with this, and hopefully one day we can have really good converged results going back to our experimental colleagues with good numbers and predict what's going to happen if you change the structure.